afternoon, sir, and welcome.
Thank you, sir.
Thanks a lot for.
Good afternoon, sir. Good afternoon, Dr. Bansal.
Good afternoon, Dr. Rashmi. Good afternoon to everybody.
Good afternoon, sir.
सर थोड़ी देर के लिए अभी स्क्रीन अनशेयर कर दें तो once we start then we can come back to this.
Sir, if sir is ready, I think we can start.
Yeah, we are 3 minutes to 3.
Screen sharing तो हो रही है क्या?
सर अभी सर थोड़ी देर बाद कर दीजिएगा अभी 1 मिनट हम लोग थोड़ी देर we will have a little introduction and then sir you can Professor Gupta sir.
Yes.
You can share the screen a little later once we start. Just
Okay.
give me 2 minutes, yeah.
अभी अनशेयर कर दीजिए सर.
Yeah, that's fine, sir. So, if you permit, sir, we can start now. It's 3 o'clock, and
please.
we have we already have 1,000 participants who have joined us. 1K you can see in the participant list.
Already more than 1,000.
Yeah.
Yes, sir.
So I'd like to extend a warm welcome to Professor P.K. Gupta, speaker of today, and many distinguished colleagues who have joined us on this webinar, Professor Bansal, and all the CAST team members. So I would like to briefly tell you about this important program funded by National Agricultural Higher Education Program, NHEP, which is supported by World Bank. Dr. Bansal was instrumental in getting it through in the last period of his service at ICR. And this program of NHEP has supported CAST, what is called Center for Advanced Agricultural Science and Technology, at many places, and IRI is one of them, with a grant of 25 crores. And the major focus of this program is unique in the sense that this is for the capacity building of students, where we identify promising students at master's level, PhD level, and they are sent to laboratories abroad for their training. And also they are supported in terms of contingency for doing their research. The grant can vary from something like 10 lakhs to 20 lakhs per student. And at the same time, we organize a large number of training program, in-house training program through this. And as a part of that, we also have started a lecture series, the present one. We had several speakers who have already presented their lectures, including your student Raji. He was here with us one day, and it's a great pleasure to have you on this program, sir. And friends, you all know, particularly the students, more than 1,000 who have joined, and also we have live streaming on YouTube. So on YouTube, we have another maybe 1,000 plus participants who are with us. And this is a great pleasure for us to have Professor Gupta with us, who is the most outstanding researcher and teacher that we have in the field of genetics, cytogenetics, molecular breeding, population genetics, any branch of genetics that you name. Professor Gupta's eminence is quite obvious from the fact that his contribution to genetics through his writings is enormous. Most of us, almost all of us, have read his books in genetics and cytogenetics and biotechnology at one or the other stage. And the beauty of his writing is that he takes the most complex subject and expresses it in the simplistic manner which is understood by a common student. So that's the beauty. That's what I have at least enjoyed when I was doing my BSc Agriculture and MSc Agriculture. Many things in genetics and cytogenetics which you could not understand, reading advanced textbooks like Strickberger and Sabanga, it was easy to understand those concepts reading Professor P.K. Gupta. He is a fellow of all the Science Academy of India, Indian National Science Academy, National Academy of Sciences India, Indian Academy of Sciences, National Academy of Agricultural Sciences. So I have no words to describe the attainments and achievements that Professor Gupta has made. He is a role model for each one of us. It is very difficult to reach to his level. You look at his I-10 index, H index, and anywhere we cannot match him. And even at this age, his ability to spend long hours on reading and immediately responding. For this presentation, he was so concerned that he sent his presentation 3 times to me today itself. And he said, Ashok, this is improved one, use this one. And the last one that he sent is just a couple of minutes back. And I hope we have that presentation with us and that he's going to share the great topic from Mendelian genetics to advanced genomics. And I got he's so critical. While typing, we made a mistake. From Mendelian genetics, we wrote it as Mendelian genetic. And then I got a reply, Ashok, the title is wrong, correct it. So we had to correct it and again air it. Thank you, sir, so much for agreeing to our request, and we look forward to listen to you. Thank you very much, sir. Professor Gupta. So you can unmute yourself, sir, and then.
Yes, Ashok and dear friends, it's a great pleasure to accept this invitation to deliver this lecture. Ashok is so fond of me. I always have superlatives to talk about what I have been doing during the last about 60 years now. And I must tell you, I enjoyed every moment of these 60 years, learning and teaching and writing various aspects. So when I received this invitation, initially I thought that I will talk something about the research we are doing, but then Ashok desired that I should have a very broad subject which should be useful for students starting from BSc and to the teachers who teach these students. And therefore I selected this topic, which is a very broad topic, and you will find that although most of the areas which I will talk about, they are elementary genetics, but in every slide I will tell you something about what has happened in those areas in recent years, including even Mendel's laws. So we start from Mendelian genetics. You will see that I will start from Mendelian genetics, starting even before Mendelian genetics started in 1900. I will also talk about the biometricians in 1980s, Francis Galton and other work which was done during 1980s. And then after the first 10 slides, then I will pass on to the next. There are different eras. And genetics, as you know, has grown during the last 100 years or 150 years at an exponential rate. And I don't think any science has really grown at that fast speed. See, this is the figure which I have taken from the internet. There are 7 traits which Mendel utilized. And in these 7 traits, Mendel analyzed the data which he obtained and proposed the 3 laws the law of dominance, law of segregation, and law of independent assortment. And he described, and these were rediscovered in 1900, but you need to know more about it, because for all these 7 traits, we do not know the molecular basis, and later on molecular basis was determined. And this slide I have taken from one of the publications where the molecular basis for at least 4 of the 7 traits has been shown. The genes have been cloned and characterized, and it has been shown how these 4 characters are controlled at the molecular level. You can see that the first traits listed is round, seed, and tall and dwarf, stem length, cotyledon color, seed coat, flower color. These 4 particular traits are now known at the molecular level, and we know the genes which control these 4 traits. The other 3 traits, I do not know whether during the last 3-4 years, they have also been characterized. But another important feature which you will note in this particular slide is that the linkage groups in the 5th column, linkage groups are given, and you will note that the linkage groups are only 5 of the 7. Earlier, in the textbooks also, I have written initially in my own textbook, I wrote that the 7 traits are located on 7 different chromosomes. And that was considered to be the reason why Mendel was successful. And I remember Ashok came to my lab one day and he told me there is a mistake in my book. And then, of course, I corrected it. And you should know this is a very important observation which the students should know, that these 7 characters, the genes for these 7 characters are not really located on 7 chromosomes. They are located on only 5 chromosomes. And therefore, all the 21 combinations of these 7 chromosomes cannot exhibit independent assortment as was visualized by Mendel. You can see there are some questions about Mendel's results, which all of you should carefully observe, because this is what we normally don't teach our students. You see, one thing is that Mendel thought of complete dominance for all the 7 traits which he studied, but later on, within few years, it was observed that for many traits in all plants and animals, we have incomplete dominance. The F1 shows intermediate character. The second thing is that independent assortment, which Mendel showed, this does not hold good in all the cases. And the results which Mendel presented were available only for 2 different combinations of genes. And this is R and I and R, I and A. These are different traits which are included. And results of all the 21 combinations were not presented by Mendel. And therefore, we wonder that whether or not all the 21 combinations were really studied by Mendel to study the independent assortment, although he claimed that independent assortment will hold good for all the 7 characters which he studied. The most important combination of traits here, which is obvious, is the height of the plant, and the other character is pod form. These 2 genes, L, E, and B, are located at a distance of 12.6 centimorgan on a particular chromosome, chromosome 3, and therefore there is no question of getting independent assortment between these 2 genes, L, E, and B. And therefore, we have to tell the students that although according to Mendel, independent assortment holds good for all the pairs, 21 pairs of 7 characters which he studied, but at least in one case, LEV, independent assortment cannot be observed as examined through linkage studies later on. Another important observation which has been made by Ronald Fisher, those of you who have not heard the name of Ronald Fisher, is the father of statistics in genetics during the last century, and made very, very important observations and gave us the tools to apply statistics in genetics. According to Ronald Fisher, who re-examined the results of Mendel, he thought that the results are too good to be true, because the probability levels which Mendel obtained in his results, were more than 99% in all the cases, and therefore Ronald examined the results and found that the probability of getting for all the traits such a high probability is rather not possible, and therefore Ronald Fisher actually used the word that Mendel or his colleagues falsified the results to match what Mendel was expecting. There is still controversy. People keep on defending Mendel's results, but the controversy goes on. Another important observation which I like to make is that the 7 characters which Mendel studied were qualitative traits. Qualitative means they were discrete traits. You could distinguish the plants only in 2 groups, showing contrasting traits, and each controlled by a single gene. But against this particular assumption of Mendel, we now know that majority of traits are actually quantitative in nature. You can find continuous variation, and each trait, in my opinion, is controlled by a large number of genes. There is hardly any trait, in my opinion, which will be controlled by a single gene. Next slide will show this. The quantitative traits and qualitative traits, if you compare them, qualitative traits are discrete traits and monogenic, sometimes oligogenic in nature, and quantitative traits, on the other hand, have continuous variation and are mostly polygenic in nature. And some of these traits have been explained in the form of multiple factor hypothesis, which was proposed near about 1905 up to 1918. A large number of papers were published which tried to explain these quantitative traits on the basis of multiple factor hypothesis, which assumes only few genes, not a large number of genes. You see, coming back to 1980s, in 1980s, there are 3 important scientists who conducted research on inheritance of human traits: Galston, Pearson, and Weldon. And these 3 scientists, while conducting studies and applying correlation and regression to the results which they obtained, they came to the conclusion that the character is continuous in nature, and if you compare the traits in the parents and the children, then the traits in the children have a very strong regression on the characters which are found in the parents. And therefore, they proposed that the characters are controlled by a large number of genes, and they are quantitative in nature, and they are continuous, and therefore you cannot explain these traits on the basis of discrete traits or single genes as proposed by Mendel. And therefore, they, in a way, challenged the hypothesis of Mendel that individual traits are controlled by single genes, and that they are not quantitative in nature, and that they are discrete in nature, and you can understand and study the dominance in segregation and independent assortment between these T genes. Then Ronald Fisher, who is the father of statistics in genetics during the last century, he provided reconciliation between Mendelians and biometricians. Galton believed that there is continuous variation and natural selection, while Mendel believed, and Mendel and Bateson and his followers believed, that there are discrete genes and you can explain them on the basis of Mendel's laws. And it was possible for Ronald Fisher to reconcile these 2 ideas, and he suggested that quantitative traits or inheritance traits which were studied by Francis Galton, they could also be explained on the basis of Mendel's laws. Then he was the person who also in 1936 published a paper where he described that Mendel's results were too good to be true. The probability of those 7 pairs of traits which are obtained utilizing chi-square, they were all more than 99%, which is very unlikely if you conduct experiments. Another hypothesis which was given by Francis Galton was eugenics. On the basis of his studies, he proposed that intelligent people should be allowed to give birth to more children, and those people who have low intelligence, they should be discouraged to produce children so that the human race may improve. This eugenics kept on surviving, and at certain places this was also practiced, but ultimately for ethical reasons this was given and opposed in all parts of the world. And I feel that in China to some extent eugenics is still practiced. Ronald Fisher also gave analysis of variance, how analysis of variance can be done, likelihood, infinitesimal model he proposed. According to infinitesimal model, those of you who never heard about it, according to Ronald Fisher, every character is controlled by a large number of very, very small genes. This model, of course, Ronald Fisher does not hold now, because we know all quantitative traits are controlled by some major genes and a large number of minor genes. They are not all controlled by small minor genes. This is a very important figure which I have liked. I learned about this figure only recently. You see, one of the associate of Francis Galton, Weldon, published a very, very important paper in 2002. It was a very long paper, about 100 pages. And this was published in Biometrica, which was the journal which was started in 2002. In the first issue of the first journal, Weldon published this paper where he collected the seeds of pea from the market and demonstrated that for every trait which Mendel studied, there was a continuous variation available if you collect large germ plots. Therefore, in a way, he demonstrated that the characters which Mendel studied were really quantitative in nature. And if you study them together, now you will find that a large number of QTLs, major and minor QTLs, control these traits. So they are not really discrete. Mendel selected those discrete phenotypes only to explain a principle or to understand the basis of inheritance. But that does not mean, even today, you can always select plants with discrete phenotypes and explain those discrete phenotype differences on the basis of individual genes. But that does not mean that there is no continuous variation available. When you collect all the phenotypes and then try to study the complete variability which is present, you will find that almost every character is controlled by continuous variation, and there are a large number of QTLs of genes which control these traits. This is again about eugenics. I mentioned about it. Eugenics is the practice of improving human species by selective mating people with desirable hereditary traits. It aimed to reduce human suffering by breeding out disease, disabilities, and other undesirable characteristics from the human population. Eugenics was initially practiced in many countries but could not be continued for ethical reasons. In the previous slide, I mentioned that to some extent this is practiced in China even today. By saying that, I meant that in China there are certain rules that every lady who is pregnant will have to undergo tests to find out whether the fetus contains any very dangerous diseases. And there is a law that if the disease is uncurable, then the lady has to undergo abortion. This is a compulsory. This is one way of practicing eugenics in a way. But nevertheless, eugenics has been discontinued in all parts of the world for ethical reasons. Then this is explanation of Mendel's laws on the basis of chromosome theory. Chromosome theory means that genes studied by Mendel are located on chromosomes. There is another theory for the students. I always like to distinguish between chromosome theory of inheritance and chromosome theory of sex determination. Chromosome theory of inheritance means that genes are located on chromosomes, and chromosome theory of sex determination means that XY chromosomes are responsible for determination of the sex. This chromosome theory of inheritance was proposed soon after Mendel's laws were rediscovered by these two scientists, Sutton and Boveri, and they studied meiosis and found a parallelism between the behavior of the chromosomes during meiosis and mitosis and the behavior of the Mendel's genes during segregation. And because of this parallelism which they observed, examining this in several organisms where homologous chromosomes could be distinguished, they proposed that the genes studied by Mendel must be located on chromosomes. And this was later on approved with variety of experiments which were conducted later on. This was about up to 1910, Mendel and the multiple factor hypothesis, the science grew during that period. But then starting in 1910, we had another very important period of genetics, which has grown very fast during the next 20 years, starting from 1910 to 1925 or 1930, where a team of scientists, including Thomas Hunt Morgan and his colleagues, Bridges and Sturtevant, they had 3-4 important persons. They took Drosophila as the material because they found certain advantages in using this material. You can see these two flies, one with red eye, the other with white eye. And this was one of the very important traits which was examined by them. And by utilizing a large number of such traits in Drosophila melanogaster, they were able to prepare the linkage maps. They for the first time demonstrated that there can be genes present on the same chromosomes and they will be linked together and therefore will not exhibit independent assortment. And it should be possible by studying recombination frequencies to map them on the chromosomes. This is a diagram showing a large number of different traits in Drosophila. You can see in the top row, there are wild type traits and then the mutant type at the bottom. And by crossing these mutants, genetic maps could be prepared and the genes for these different traits could be assigned to the chromosomes separated by distances which were proportionate to recombination frequencies. This was the basis of preparing genetic maps. It was assumed that the distance between genes is proportionate to the recombination frequencies between these two genes. And on the basis of that, genetic maps were prepared, and these genetic maps only represented the recombination frequencies between genes and not the physical distances. On the bottom, you see the physical map. The physical map differs from the genetic map because recombination frequencies are not the same throughout, and therefore you find that the proportionate genetic distances are not proportionate to physical distances. And Drosophila was such a wonderful material that in case of Drosophila, salivary gland chromosomes were available, which were 1,000 times longer, and banded chromosomes were there, with the help of which bands could be assigned, and with the help of bands and deletions and duplications which were available, physical maps could be prepared in Drosophila for the first time. This is a diagram which really shows that the physical distances on the chromosomes will not be proportionate to recombination frequency. If you plot the observed recombination frequencies against the genetic distance, you will find that the dotted row here, you can see the dotted row, this shows, this is the type of curve we should obtain if there is a linear relationship between the recombination frequencies and the map distances. But actual frequencies do not map like this. The curve goes down. It means as the genetic distances is increasing, the recombination frequency does not increase at the same rate. And at 50% recombination frequencies, no further increase in recombination frequency will take place. And therefore, this figure demonstrates that using recombination frequency as a parameter for preparing genetic maps was not the true approach. And therefore, for small distances, you can use this assumption that observed recombination frequency is directly proportional to the genetic distance. But for longer distances, this hypothesis or assumption was found to be wrong. And therefore, following this, mapping functions were prepared. Many students are not familiar with this. These mapping functions are based on Poisson distribution in statistics. Poisson distribution is a distribution which is utilized for explaining distribution of particular traits or particular factors which occur in very small frequency, very small frequency. So meiosis also between two genes occur at a very low frequency. And therefore you can see here that if this is the physical length, this physical length may be expressed by a longer distance if the recombination is low, and it can be represented by a smaller distance if the recombination frequency is low. So the recombination frequency in different parts of the chromosomes are not proportionate to genetic distances, and therefore it is necessary for you to find out a method with the help of which you can prepare the genetic maps which are really true genetic maps and not the maps which are based purely on recombination frequencies. The first such mapping function was proposed in 1919 by Haldane. He prepared this particular function where recombination frequencies can be converted into map distances. But this Haldane mapping function also was not found to be most appropriate because of the fact that Haldane did not take into consideration negative interference. You see, a particular region of the chromosome, if recombination is taking place in a particular region, it interferes with recombination in the adjoining regions. This is called interference. So this interference, which is of common nature, was not taken into consideration by Haldane, and therefore a more appropriate mapping function was proposed by Kosambi, and there are at least another 5 or 6 mapping functions which are available, but most of the geneticists must be knowing that we use throughout the world. Kosambi's mapping function is the function which we always utilize, because Kosambi's mapping function takes into consideration interference also. So it has been shown as shown in this particular diagram, recombination rates differ in different regions of a chromosome. This has been shown. This has been shown even by physical distance, as one of the speakers came to Meerut to deliver a lecture on this. He showed us beautiful slides where he demonstrated physically that recombination frequencies really differ. Recombination rate does not proportionately increase with distance between genes. This is another observation which was made, and therefore these Haldane mapping functions and Kosambi mapping function became available, and our Kosambi mapping function is utilized now everywhere in the world for preparing genetic maps. Then, after this period of recombination and preparation of genetic maps, starting from 1910 to about 1925 or 30, these were 20 years of very strong and active area of research on recombination and preparation of genetic maps. But then there was a period when it was realized that you don't have sufficient variability available in nature to study the genetics of all the traits. And therefore, if you don't have the variation for a particular trait available in nature, how to study the genetics of this particular trait? And therefore, it was considered necessary to create variation artificially. And H.J. Muller was the first person who in 1927 demonstrated for the first time that X-rays can be used for inducing mutations, for which he was later on in 1946 awarded Nobel Prize. So you can see that starting in 1927 or 30, there is a period of about 40 years, I will say, starting from 1930 to 1970. There was a very active period of induced mutations. You know that IARI also, Dr. Swaminathan led a group, then H.K. Jain also. So there was a very active group in IARI also, where induced mutations were studied at large scale in many parts of the country, including our own lab. We also published some papers. We also created a variety on the basis of induced mutations in mung bean, and such varieties were produced in IARI also in pulses and other crops on the basis of mutations. And some of you must be familiar that these varieties produced by induced mutations are registered in international atomic energy agencies. They publish a bulletin, and more than 3,000 varieties are now known to have been produced on the basis of induced mutations. So induced mutations played a major role, not only in the study of genetics, but also in crop improvement programs. This is another very important picture, Auerbach. See Auerbach, and while showing this slide, I recall our good friend V.L. Chopra. He studied under her supervision on chemical mutagenesis in University of Edinburgh, and she was the lady who for the first time demonstrated that not only irradiations, physical radiations, but also chemicals can be used for inducing mutations. And the chemical which she used for the first time was the mustard gas. And because she was working at the time of World War, she postponed the publication of her results for a few years with the fear that mustard gas may be used against human beings, and therefore she deferred publication of her results to about 1947-46. And then, of course, she utilized other chemical agents also for mutagenesis, and she is the most important person on chemical mutagenesis in the world. And she later on published 2 books also. I have them in my shelf, and I like to read those books written by her. You see, this was followed. You see, when you are studying the morphological traits, like those studied by Mendel, like those studied by Morgan and his team, and also like those studied by Muller in irradiation of mutations, these are traits which are easy to handle and easy to study, easy to use for demonstration. For example, in case of maize, there are a large number of traits, and IRI has produced a bulletin on maize, how maize can be used for teaching students, because you have large number of mutants available which can be shown to the student. You can also show the segregation of different traits utilizing the example of Mendel maize. But the study of these mutations of phenotypic traits, it was difficult to study the molecular mechanism which is involved. And therefore, Beadle, who was initially working on Drosophila and Neurospora, he worked in Drosophila using eye transplantation experiments to find out if you transplant one eye into the other, what changes, what happens. But he was frustrated with both these systems, Drosophila and Neurospora, and therefore he shifted to Neurospora for the study of biochemical genetics. And starting in 1940, there were about at least 20 years of period of biochemical genetics where biochemical mutations could be studied, utilizing, in case of Neurospora, you can see that spores were irradiated with the help of X-rays, and some spores have random mutations, and these are studied on complete medium. All of them will grow. On the minimal medium, the mutants will not grow, and the mutants which will not grow on the minimal medium, they can then be tried on supplemented media, and you can find out the deficiency which the mutation has caused. And then it can be studied further. So Beadle, utilizing this kind of mutations in case of Neurospora, was able to study some of the biochemical synthetic pathways. And he suggested the theory of one enzyme, one hypothesis, one gene, one enzyme hypothesis, which was later on converted into one polypeptide, one gene hypothesis. So this work on biochemical genetics, which continued for at least 20 years, gave useful information and will help in deciphering the biochemical synthetic pathways for many, many traits. For example, even in case of Drosophila I, biochemical synthetic pathway could be deciphered utilizing some of the mutations. Another very important discovery in 1940s on the genetics, this is the genetics aspect, we are not talking, still not moving to the molecular level. You see, in 1940s, there were two scientists, published by them. They demonstrated for the first time, you see, according to Mendel, the gene is a point on chromosomes. And we often teach our students also that gene is a point on chromosome separated by distances which are proportionate to recombination frequencies. And there was no question of recombination taking place within the gene. Intragenic recombination could not be thought at that particular time. I remember in 1958 when I started teaching genetics, I was shocked to see a chapter in Sinnott and Dunn's book where it was shown that intragenic recombination can take place. Intragenic recombination means that there are different alleles, they are not at the same location, they are present at different locations within the gene, and you can map them and you can find out recombination between them. And on the basis of this was later on from Drosophila, this study was extended in case of Phage, R2 locus, rough plaques. These are rough plaques, these are smooth plaques. So this locus of R2 locus, mutant which was studied by Benzer, was found, he studied about 400 different mutational sites in this particular locus, and intragenic recombination between these mutational sites were studied. And on the basis of this, he classified this locus R2 locus in two cistrons. I want to spend a little few minutes on this cistron because I've seen that even many teachers do not know what is a cistron. Cistron, when I ask teachers and students, they say it is a functional unit. Yes, it is a functional unit. That is not incorrect. But why do we call it cistron? That is the question I ask. And the teacher should be able to explain why do we call it cistron. We call it cistron because the two alleles present in the same cistron, if present on different chromosomes, will not give you the wild type. This is complementation. So the two alleles within the same cistron will not complement, while the two mutants or alleles present in different cistrons will give you the wild type. Complementation. This is called complementation test. So the complementation test, when you exercise, within the same cistron, you will not find complementation, and between the cistrons you will find. So you conduct a cistrons test, and on the basis of the cistrons test, you find out what is a cistron. And I think the teacher should explain it to the students. Why do we really call it a cistron? Beginning of molecular biology in 1946, I consider that molecular biology began in 1946 with the discovery that DNA is the genetic material by the experiment, classical experiment conducted by Avery et al. 1946. This was followed by experiment by double helix model in 1953. This was discovered by transfer RNA in 1958, then discovery of messenger RNA in 1961, and then genetic code 60 to 65, and synthesis of East alanine. You can see that this period, particularly the period is starting from 1953 to 1970. This was such a productive period in molecular biology. I remember when I used to teach this, one of the examiners asked this question, discuss that 1960s was the most fertile period for the growth of molecular biology. So it was such an important, so many Nobel Prizes were awarded on the work which has been done by these different scientists. Coming back to this first of these, DNA is the genetic material. This is a classical experiment which was conducted in 1928 by Griffith, transformation experiment. In the transformation experiment, Griffith took pneumococcus strains, two types of strains, rough strain, which are non-virulent and cannot cause the death, a smooth strain, which is virulent and will cause the death of the mouse. But then, if you use heat-killed smooth strain, this one is smooth strain, which is virulent, this does not cause the death of the mouse. But if you mix this heat-killed smooth strain with the rough strain, living strain, then you see that the mouse dies, which means that the non-virulent rough strain got converted into a smooth strain using the heat-killed material from these smooth strains. So a classical experiment which shows that you can transfer the genes from the culture medium or from the solution to the organism, leading to change in the genetics of that particular trait. Heat-killed strain does not kill mouse, but when used with live non-virulent rough strain, it kills mouse, suggesting that rough strain changes to smooth. Transformation principle was later on worked out. Next. This slide shows the transformation. The same experiment which was conducted by Griffith in 1928 was repeated by Avery et al. These are the three scientists, Avery, MacLeod, and McCarthy. They used the same material, but in this material, they conducted an experiment where the solution of the heat-killed strain was treated by proteinase for killing the protein, by ribonuclease to destroy the ribonucleic acid, and then by deoxyribonuclease to destroy the DNA. You can see that when there is no DNA in this particular case, when you destroy DNA, then the convergence transformation does not take place. But in these two cases, when you destroy protein or RNA, then the transformation takes place, suggesting thereby that neither protein nor RNA is involved in transformation, and it is only the DNA which is involved in transformation. So this was the first evidence available which showed that DNA is the genetic material, because if you have read the history, you will find that before this there was a controversy whether proteins carry the genetic material or the nucleic acids carry the genetic material. Another experiment was conducted by Hershey and Chase. This young lady, Martha Chase, was responsible for conducting this experiment with Alfred Hershey. And they took radioactive substance available in bacteriophage T2 phage and labeled the protein and the DNA by radioactive substances. And with the help of using these radioactivity labeled protein and DNA, they demonstrated for the first time that only the DNA enters the cell and is passed on to the progeny and not the protein. The protein is labeled by sulfur, but no sulfur radioactivity is found in the progeny. In the other hand, if you label it with phosphorus, which is present in DNA, then you find that in the progeny, radioactive phosphorus is available. So this was another experiment which demonstrated that DNA is the genetic material. There are other experiments also which I am not focusing. For example, tobacco mosaic virus was also used to demonstrate that RNA is the genetic material and not the DNA. Then Watson and Crick's model 1953, the double helix model, Watson and Crick. I have written in my book about Rosalind Franklin's contribution, and we often talk about it, that major work which was utilized by Watson and Crick to propose this model was done by Rosalind Franklin. Her contribution using X-ray diffraction images was recognized only posthumously, and therefore she was not a part of the Nobel Prize which was awarded to Watson and Crick. This is the X-ray diffraction picture which was used for proposing the double helix model. Then in the same, during 1950s, transfer RNA was proposed, 1958, by Zemechnik and Hoagland. They proposed that transfer RNA is involved in protein synthesis, that amino acids are carried by transfer RNA. And then this transfer RNA was studied in detail by Robert Hawley in 1965. This sequence was proposed, and clover leaf model was proposed by Hawley. And Robert Hawley was awarded Nobel Prize with Khorana and Nirenberg for this particular work. And then later on, this clover leaf model was proposed on the basis of the sequence, but crystalline form of this transfer RNA was not available at the time. And when crystalline form became available, then three-dimensional structure could be studied by Arthur Klug, who also later on got Nobel Prize. And on the basis of this crystalline structure, this L-shaped model for transfer RNA was proposed by S.H. Kim. So you can see that transfer RNA is a small molecule, but is a wonderful molecule. It performs important functions during protein synthesis and was utilized for a variety of works. And you should also know that this was one of the first RNA or DNA molecule which was fully sequenced and therefore was utilized by Khorana for synthesis of gene. You can see on the basis of the work done during this period and then discovery of RNA by number of scientists, messenger RNA, Brenner, Pardee, Jacob, Watson, this model of protein synthesis was proposed in 1961, where transfer RNA carries the amino acids, and then with the help of these amino acids and this RNA, this ribosome, the protein synthesis takes place. Then 1960, I still remember when I started my career at Gorakhpur University, there were headlines in the newspaper, genetic code cracked. And I didn't know what does it mean at the time. So genetic code was cracked in 1960 for the first time by Nirenberg and Mathai, and they used artificial triplets which were utilized in order to find out what kind of amino acid they would carry. And then Khorana, of course, he synthesized DNA molecules and of known sequence and utilized them to find out which triplet codon codes for which particular amino acid. So within 5 years, that was rather too fast. Our complete genetic code was deciphered within a period of 5 years, 1960 to 1965, by Nirenberg and his team and by Khorana and his team. They were awarded Nobel Prize in 1968 for this work. I told you that East alanine transfer RNA sequence was discovered by Robert Hawley in 1965. Very, very important discovery. And immediately when this sequence was published, Khorana took this sequence and started synthesis of the gene for this transfer RNA, which is about 77 bases long. And by synthesizing oligonucleotides brick by brick, he ultimately was able to synthesize this particular transfer RNA, which is 77 bases long. And by the time he completed the synthesis in 1970, he came to know that this gene is an incomplete gene because this was the ultimate product, but the gene which is responsible for this gives a much longer precursor transfer RNA molecule, which ultimately gives rise to this. And later on, therefore, Khorana started synthesis of the whole gene and synthesized the gene for this particular transfer RNA, which was about 126 or maybe more, about 200 bases long. Another period starting at the end of 1960s, 1968, 69, is the unique and repetitive DNA. For the first time, we learned that there is large fraction of DNA which is present, which is of repetitive nature. See, when the frequencies of mutations were compared with actual mutations and the genome size, the total DNA which is present, there was a lot of anomaly which was found, and the rate of mutations could not be explained on the basis of genome size and the rate of mutation frequencies. And this led to the discovery of repetitive DNA. You can see that this DNA is of a eukaryote like hexaploid wheat, where you have a repetitive fraction and you have unique fraction here. And this is actually a reassociation kinetics. You deassociate DNA, denaturate DNA, and then reassociate. And this curve shows the rate at which reassociation takes place. Repetitive fraction will have many copies, and therefore reassociation will take place faster. Unique fraction will have less number of copies, and therefore reassociation will take time. And this is described in form of Cott curves. And these curves were published in large number during that period. And I remember one of the very important paper published from India was repetitive fractions in case of wheat in 1973 by Chitranjan Bhatia. And I still remember when 1974, I visited UK, everybody was asking me about who is this Chitranjan Bhatia? How could you do this work in India? So you can see that this was a very, very important subject, study of repetitive DNA. And most of the time we thought at that particular time that this repetitive fraction, what is it doing? Whether it is all nonsense, sometimes also we call it nonsense DNA. And therefore for almost 10, 20 years, we did not know the function of this repetitive fraction, which is clearly understood now. Earlier we thought that the repetitive sequence is not transcribed and therefore does not take part in any function. But later on we found that all repetitive, most 99% of the DNA is transcribed, although only less than 1% contains genes. And therefore rest of the DNA remains in RNA form and takes part in regulatory functions. And this is an important area for study. Many labs in our country, including our own lab, is trying to examine the role of non-coding RNA, which is available in large number of different types in all organisms. Another important person, those of you who are not familiar with this name, Frederick Sanger, who is the only scientist who got Nobel Prize twice. There are other Nobel Prize winners who got Nobel Prize twice, but he is the person who got it twice for science. The first Nobel Prize he won was for studying the sequence of insulin, which has a chain with 21 amino acids and B chain with 30 amino acids. And then in 1980, he again got a Nobel Prize for studying the DNA sequence of 5x174 with circular 5638 bases long. So you can see that development of techniques, Sanger was a marvelous person. He developed the technique for DNA sequencing in early 1970s, and that technique of sequencing, dideoxy DNA sequencing, was continuously used for at least 30 years by all over the world before we came to the next generation sequencing. Another important discovery during 1980s was split genes. Nobel Prize winning discovery again, Richard Roberts and Philip Sharp. They discovered that the genes are not continuous sequences. There are coding sequences and there are non-coding sequences. The coding sequences were called exons and the non-coding sequences were called introns. And all these sequences are perhaps transcribed, and intron sequences are spliced out later on to give rise to the sequence which take part in translation. Very, very important discovery. I remember in 1982-83, I went to the library and the cover page of Nature shows split genes. I was shocked at that time, and then of course this led me to read about it. Another important era, I would call it, started in 1980 with RFLPs, molecular markers, for the first time. You can see one of the drawbacks of the classical genes and alleles, which we used for genetic studies, was that you did not have enough markers available, alleles available in order to conduct genetic studies. And therefore, the genetic maps could not be prepared. Many traits could not be placed on the genetic maps. And therefore, that being a major limitation, the availability of molecular markers in 1980 and onwards, during the last 20 years, this has been such a productive and fertile area of research that many of us in the country and outside got involved into this. I was very lucky to learn RFLPs in Canada with one of my friends there in 1985-86. That helped me also to develop a lab in Meerut University, where molecular markers have been continuously used for the last about 25 years. And students like Rajiv Varshne and Manoj Prasad at NIPGR, they are the products of that particular projects which we had. And I'm thankful to Dr. Chitranjan Bhatia, who in 1995 invited me to Delhi and asked me that I should establish a lab for molecular biotechnology in case of wheat, and he would give me anything I want. And that was the beginning of a new career when I was about to retire in 1995-96, which during the last 25 years kept me busy. You can see on the basis of molecular markers, particularly in case of grasses and cereals, maps could be prepared and synteny could be studied. The same molecular markers are present in all grasses, and there is a synteny. Synteny means same markers present on chromosomes of different grasses, and collinearity means that they are present in the same linear sequence in which they are present in one species. So both synteny and collinearity was studied in different grass species, and Mike Gale from Norwich, he prepared this particular diagram for 7 grass species, which is a very, very popular figure and utilized by many of us in delivering lectures. Molecular markers during 1980 to 1990, they were utilized for preparing some maps and were very useful. I also published 1 or 2 papers during 1980 to 1990 on ribosomal DNA, etc. But then in 1991, there was a paper published by Lender and Bookstein from Harvard, which was a very, very important paper published at the time. QTL internal mapping was suggested because by molecular marker, you can find out the markers associated with a particular gene, but you cannot really find out the position. Therefore, QTL internal mapping was suggested in 1991 by Lender and Bookstein and was extensively utilized during the last 30 years for all crops and human beings, and also in case of livestock. And another technique was proposed by Ed Buckler, association mapping, that was a little later, about 10-15 years later, early years of 2000 or 2005. Association mapping, I remember, it started actually in 2000 or nearly 2000, because in North Carolina State University, I attended a summer course where there was a lecture on association mapping, and I knew nothing about it at the time. So you can see that from 2000 onwards, and after returning from North Carolina State University in 2000, I asked my student Joy Roy, who was writing his thesis, will he like to conduct association mapping? And he readily agreed. His thesis was delayed by 1 year, but he was one of the first person from our lab who published a paper on association mapping in Current Science. And this association mapping can have 2 different types. The first studies which were published on association mapping involved candidate gene approach, not the genome-wide association studies. You will realize it. Surprisingly, we normally conduct genome-wide association studies only now. We hardly conduct any candidate gene approach. But the first approach, the first experiments which were conducted in the beginning was candidate gene approach. Next slide will show this. You can see linkage versus association mapping. You see the QTL internal mapping is sometimes described as linkage-based study. Linkage, because we find out recombination frequencies here in order to find out the position of QTLs. And association mapping, on the other hand, is described as LD-based, linkage disequilibrium-based. You see, the QTL internal mapping, we utilize the map which is prepared on the basis of linkage, and in association mapping, we do not use such a map. We do not use such a map, and therefore the associations which you find in case of internal mapping is based on linkage. Association mapping which does not make use of any map is based on linkage disequilibrium. Linkage disequilibrium means that the two genes or two markers are inherited together with irrespective of the linkage between them. Sometimes they can be inherited together even if they are not very close to each other. And that is one of the serious drawbacks of association mapping, that it is based on LD, and LD is not always based on linkage. Although in linkage studies, initially biparental mating was used, but later on large number of multi-parental populations were also used. Magic population is one of the popular multi-parental population, multi-parent advanced generation intercrossing. These magic populations are also being used in rice and many other crops now, in wheat also. This is one of the diagrams from one of our first paper published on interval mapping, Lander and Botstein, and you can see that this is the output you get from the computer. I remember I brought the QTL cartographer from North Carolina State University in 2001, and Joy Kumar Roy spent the whole night in order to install it and run it. Now it takes not more than 5 minutes to get this output. So that is the beginning of QTL internal mapping in our lab in 2001, when I came from North Carolina State University, and Joy Kumar Roy was the first person who installed this QTL cartographer and gave the first output from this. Association mapping can be done in two different ways. You can see, I told you earlier also, there is genome-wide association mapping and also the candidate gene approach. You can take the whole genome, you can do the genotype of the whole, and of course, the germplasm will be phenotyped. Phenotyping will be done in both the cases, but genotyping will be done in case of GWAS or genome-wide association mapping only for the whole genome, but in case of candidate gene, we prepare the genotype only for a fraction, only for the region in which we are interested. And utilizing these two different approaches, hundreds and thousands of studies have been conducted during the last 15, 20 years. Association, yes. Two approaches. Now these approaches of marker-assisted select, this QTL internal mapping and association mapping were conducted particularly with a view to determine and identify the markers which can be utilized for marker-assisted selection. And marker-assisted selection, for those who do not understand it, means that you can select desirable plants without studying the phenotype in which you are interested. You can select the plants at the seedling stage for protein content which you don't determine, and therefore it facilitates your work, and therefore large number of studies have been conducted using marker-assisted selection. Ashok himself was involved in developing a large number of rice genotypes utilizing marker-assisted selection, and I always quote those examples of IRI and other places, even in my book, where I write how mass, even in India, has been utilized. H.S. Gupta was another person who utilized this particular approach for developing QPM, quality protein maize, Vivek. Mass for complex traits. This is a question which I always keep on talking about. You see, majority of marker-assisted selection, where we utilize markers for marker-assisted selection and plant breeding, they are all simple traits. For example, disease resistance is one of the most common traits which has been utilized. In some cases, drought resistance has also been utilized. But majority of complex traits are controlled by few major and many minor genes with low heritability. And this has not been possible. In majority of cases, this has not been possible. And this is why you will find later on in my presentation, I will talk about genomic selection, in which you consider all the markers which are mapped in order to find out the desirable plants. And then marker-assisted recurrent selection was proposed, genomic selection was proposed, which deal with complex traits, but hardly any ongoing programs in India. Now, of course, at IRI, marker-assisted recurrent selection has been undertaken. Prabhu has done it, his team, and genomic selection also is being undertaken now at several places. This slide may be a little old, but these are the two areas which will develop and will be utilized in the next 5 years. Then the next aspect is the genomics, which started in 1995. And I remember my colleague, Dr. H.S. Balian, went to attend the International Wheat Genetics Symposium in Canada, and he came back and he told me that everybody is talking about genomics now. So whole genome sequencing started in 1995. And there are two methods available for this, clone by clone sequencing and shotgun sequencing. Clone by clone sequencing needs physical mapping, as done here. This is the physical map prepared. And on the basis of physical map, you prepared a minimum number of clones which you will have to sequence, because overlappings can be avoided, and therefore your sequencing work is reduced. Your physical map preparation work is very laborious, but then your sequencing work is reduced, in this case clone by clone. But in shotgun sequencing, we do not prepare the physical map, but start sequencing small fragments in large number, maybe 10 times the sequencing work which is done in clone by clone, and then bioinformatics has to be strong in shotgun. So with the help of strong bioinformatics, you may not prepare physical map, you may start with sequencing right away and utilize strong bioinformatics in order to prepare and assemble the whole genome sequence. This is the first genome sequence of a bacteria. You can see Haemophilus influenzae, which was sequenced for the first time. This is, of course, except the phages, etc., 5x174, these were sequenced earlier also. So Fleshman RD, and whole genome sequence was published, which was as long as, you can see, 1830,000 base pairs long, and it was sequenced by shotgun sequencing and then assembling the genome. So this marked the beginning of the genomics era, in my opinion. Then after 5 years, in December 2000, this Arabidopsis genome was fully sequenced, which was a major discovery. And it was found that it has 5 chromosomes, but there are extensive duplications. You can see every chromosome has duplications in every other chromosome. So such a large number of duplicate segments are present in such a small genome with only 5 chromosomes. And after 2000, whichever plant genome has been sequenced, they have all been found to carry a large number of duplications and ancient polyploidy, paleopolyploidy. This is other major genome sequences, Baker's, elegans, Drosophila, human genome. But of course, this is a very old slide. Now we have hundreds of them which have already been sequenced. You name a crop, the genome is available. Then in India also, number of works have been taken, genome sequencing, genomics projects were undertaken, rice chromosome 11, wheat chromosome 2A, pigeon pea genome at CRISAT, and also at NRCP, chickpea genome at CRISAT, tomato genome, and then of course potato genome. Now, once the reference genome has been made available, initially you take only one particular accession in order to find out the genome sequence. Sequence is described as a reference sequence. And some 20 years ago, we could not think that we would ever have a large number of genomes of the same species sequence. But this has become possible now. After 2010, I think in large number of cases, we are trying to find out the variability within a species, within the genome sequence of a species. And this variability is enormous. This is a figure for wheat from one of my papers, which I have published. Each wheat cultivar has about 125,000 genes. Core genome consists of 81,000 dispensable and unique sequences and 40,000 pan-genome sequences. You see, if you take number of cultivars, in this case, we have taken 3 cultivars, and when we take the sequences of these 3 cultivars, total sequences are 140,000. They have a core sequence which is only 81,000, and then they have dispensable sequences which can be dispensed with, and they are marked with D. They are the sequences which are present about 40,000. So you can see that now we have entered an area of genomics where we want to study the pangenome in order to find out the variations in the genome sequences which can be utilized for plant breeding. In case of, for example, rice, 3,000 different accessions have been sequenced, and they are being utilized for a variety of genetic studies and plant breeding. Sequencing to post-genomics era. In the post-genomics era, you can see first gene sequence, Sanger's dideoxynucleotide, second generation sequencing, 454 Illumina, third generation sequencing, PacBio and nanopore sequencing, and then multi-genome sequencing, genome versus pangenome. So all this, you can see that starting from 2005, this next generation sequencing started because we wanted to get the sequencing done at a very cheap rate. And because of the competition, it has become possible. You can get the sequencing done at a very cheap rate now. And these two techniques, which have been utilized in case of wheat, also PacBio sequencing and nanopore sequencing, they are being utilized now in a big way because they can give you long sequences. PacBio can also give you about 1,000 base pair segments fully sequenced, and nanopore, of course, there is no limit. Then another technique was proposed in 2011, genotype by sequencing, where we can sequence a large number of DNAs together by tagging DNAs with a label each time so that the sequencing becomes much easier. And this was being done in large number of cases. Now there are a number of vendors available who will conduct the genotyping by sequencing for you at a very cheap rate. Genomic selection is another very, very important area. I told you earlier that because every gene is controlled, every trait is controlled by a large number of genes, few markers will not help you for complex traits. And therefore, in 2001, this technique was proposed, genomic selection, and it has recently been reviewed 2014, and it is still in a stage when it is not being extensively utilized. We are still trying to find out to what extent and in what measure genomic selection can really be utilized for plant breeding. Key components of genomic selection: selecting a good training population, a prediction model has to be there to give this is the value. Genomic estimated breeding value has to be calculated, which will be utilized for selection. This is the scheme for developing the training population, and then this is the breeding population. So you determine the genomic estimated breeding values utilizing the training population, and then utilize it for the breeding population. This is another important slide, which not many of you may be familiar, but I came across it to study the three-dimensional structure within the chromosome. You see, these are chromatin fibers, and these chromatin fibers are folded in a variety of manner. And we talk about transregulation. You see, one gene present at a particular place can control the expression of a gene which is thousands of base pairs and kilobase pairs away from that particular sequence. Not only that, the sequence which is present in one chromosome may be regulating the sequence of another chromosome. And therefore, it has been suggested that 3D structure, the manner in which the chromosomes are folded, should be determined, and number of approaches for this have been suggested during the last about 10 years. Three-dimensional genome structure of a single cell, even within a single cell genetics is another very, very important area, which deserves a full lecture actually, single cell genetics. Perhaps sometime I will give a lecture on single cell genetics. There is a recent paper in June 26, 2020, where single molecule regulatory architecture was captured by chromatin fiber sequencing. Genome editing is another very important area. CRISPR-Cas you must have heard. And then there are, of course, this is CRISPR-Cas result. You can see this mushroom, white mushroom, and drought tolerant vaccine maize. Then there are a number of limitations of this CRISPR-Cas technique. They are listed here. I don't have much time available to go into the details, but I have published a paper on base editing, which was published in 2019, February issue of Current Science. And I delivered this lecture at IRI and also at Harvard earlier. This base editing technique was for the first time proposed in 2016. And during the last 4 years, large number of papers and large number of base editing, they have been utilized. And this technique has already been utilized in China and Japan, but nothing has been started in India so far. You can see this is DNA adenine base editors. You can read about it published in Nature. Then there is another rice base editors have been developed in China. Large number of rice base editors have been developed. This is base editing actually being done for acetyl-lactate synthase for this herbicide, for developing herbicide resistance. That is also in China. Then synthetic genomics is another very important area, which I don't have time to go upon. But you can see that in case of yeast, every chromosome is being synthesized. And all these 16 chromosomes, you can see, have been assembled together in one chromosome or in 2 chromosomes. This is an area, I published a review article on this in Current Science some time back. So in summary, we can say that genetics to genomics is an exciting journey. Mendelian and Morganian genetics prevailed during 1900 to 1940. Biochemical and bacterial genetics flourished during 1940 to 60. Molecular era started in 1970 and continues even today. Quantitative genetics started in 1990 and continues. And whole genome sequencing and synthesis started in 1995 and continues even today. I enjoyed talking to you. I enjoyed learning all this and talking about it. I thank you very much for your patient hearing. We'll be pleased to take questions. Hello, Ashok. Is that right? Okay, okay. अच्छा अच्छा गौतम पे जाऊं क्या क्या है तुम्हारा यहाँ तो रहा यहाँ तो शो कर रहा Ashok, can you hear? Kailash, can you hear me?
Yes, sir. Yes, sir.
Yes, sir. Please go ahead, sir. Yeah.
Okay.
But
Okay.
your
Slides, sir.
share, sir, you have to share slide
You
again.
need the slides?
Yeah, slide, you have to share again. Slide is not shared at this point of time. Yeah, it's coming up. Yeah, it's there now. So you can go back to 2-3 slides maybe. We were at herbicide tolerance in maize. Yeah, this
No,
was, yeah.
I didn't. Oh, in the last few slides, I lost connection.
Yeah. So you can
Okay.
start that herbicide tolerance in maize you were talking about in the last, yeah, the 59 slide, 59.
Yes. So you can see that
Slide show, sir.
What-
Slide.
what I was trying to emphasize is that this base editing approach, which I am fond of delivering a full lecture, has been picked up very fast. At Harvard, it was developed for healthcare, human healthcare. And the David Louis, who created this particular technique, he he created a company for health services. But then this technique was taken up by China and Japan in a big way, and they have already utilized this technique for creating this one important example of developing herbicide resistance in case of maize. There must be other examples also, I'm sure by now. But I have not seen anybody starting this work in India. Then this is another important area of research. I remember 2 or 3 years ago, I published a paper in Current Science when the first yeast chromosome was fully assembled by synthesis of fragments. And then later on, during the last 3-4 years, there are at least 7, 8, or 10 chromosomes which have been fully synthesized. And in 2018, there were 2 papers where all the 16 chromosomes of yeast were assembled in 1 chromosome or in 2 chromosomes. So this is an area of research which will flourish in future, because while synthesizing these genes, you can eliminate large number of genes which are not really essential. So by determining which genes are important, which are not important, you can assemble a genome with much smaller size, which is fully functional. This has been demonstrated in case of microbes also, and this is being demonstrated in case of yeast also. In yeast also, there are a number of different types of genomes which have been assembled by deleting and adding sequences. And this is, of course, the conclusion. Genetics to genomics is an exciting journey. Mendelian and Morganian genetics flourished in 1900 to 1940. Biochemical and bacterial genetics flourished in 1940 to 60. Molecular era, genome synthesis, 1970 onwards. Quantitative genetics, 1990 onwards. I have actually written a book on quantitative genetics, which should be out next month. Whole genome sequencing and synthesis, 1995 and onwards. So friends, as I told, perhaps you have not listened, I have enjoyed delivering this lecture. I have enjoyed learning this subject during the last 60 years of my career. And I am grateful to Ashok, who is so fond of me, always likes to speak about me in superlatives. But I always thank him because he invites me almost every year for delivering such lecture. It will be a pleasure for me to take any questions.
Thank you, sir. If you can unshare the screenshot, then we'll have the question and answer session.
Share.
So, you unshare it, sir. Yeah, this is fine now. So Gopal has the responsibility of taking the questions which are there, large number of them in the chat box. And the panelists who can be seen, and I'm also happy to say that Professor V.S. Dillon has joined us, and he has been all through from the beginning. In fact, he was earlier in the attendee group, and then we upgraded him to panelist category. Thank you, sir, so much for your keen interest and in the science that has always been there, and Professor Bansal and all my colleagues. Gopal, I think we can take a few questions that you have with you in the chat box.
Yes, sir. Good evening, sir.
These are the questions which have been asked by the attendees, and attendees
Yes.
are the category of viewers who cannot be seen on the screen, but they are watching and listening you. So they have put their question in chat box, and Gopal will read the question, and if you...
Gopal, sir.
Good evening, sir. Yeah, good evening, sir. And you said superlatives failed because the maximum number of participants which we could have was only 1,000, and we have exceeded that limit already. And there are around a couple of interesting questions, sir. Some of them I would like to put forward to you so that and also we will have some live questions also from some of the participants. One of the questions.
Oh, fair again. क्या connection है? What is photo level?
Sir, yeah. One of the participants, Naveen, has asked how many different types of mapping are possible. Like
Different types of mapping.
No, the
hello.
question- You see, the mapping is genetic and physical mapping. I don't understand the question.
Okay, sir. Yeah.
Number of software is available.
Yeah,
Software has-
possibly. Yes, sir.
Mapping is only genetic and physical mapping, then genetic mapping to fine mapping.
Okay, sir. One Roy has asked what should be done after identification of stable mutant. Why yield cannot be increased by mutation breeding? That is one of the questions.
No, there must be examples of yield increase by mutation breeding. I think in Moogmin, the variety which we released from Herat, it was released on the basis of yield only.
Yes, sir.
So,
There are several examples, I suppose. Yeah.
there
Sir,
are-
one question from Kumarasamy. He asks, what is the future of CRISPR-Cas technology in India?
Oh, CRISPR-Cas, I think some of the labs are already using it. And I think it will be, its use will increase in India. There is no doubt about it. NABI has published perhaps 1 or 2 papers. And there are other labs which are taking up. But I would advise them to take up base editing. Instead
Dr. Vishwanathan is already working on genome editing in IRI, actually.
Yes, I know. I know IRI people are doing it.
Yes, sir. Sir, we will have some participants who want to ask live questions. I will connect to them. There is Ankala Sai, who has raised who wants to ask a question. Ankala, please go ahead. Hello. Sorry. Ramanpreet Singh wants to ask a question, sir. Yeah.
Can I see?
Ramanpreet, yeah, Ramanpreet, kindly go ahead with the question. Hello. You have to unmute yourself.
And then they have to speak.
Yes, Ramanpreet. Yeah. You have to unmute yourself and then ask the question.
So I think there is connectivity issue.
Connectivity. Yes, sir.
There is one question, as I can see from Prakhar Singla. What exactly is the use of non-coding sequences which are transcribed?
Oh, that is a full lecture.
Full lecture.
Full lecture. Actually, because there are so many different types of non-coding RNAs. I have written a few articles and we are working extensively on this area. So what is the use? It's mainly regulatory. If she wants one sentence answer, the most important function of non-coding RNA is the regulatory of gene expression.
So in that context, sir, if I may ask that when we talk of synthetic genomics and we are saying that we will assemble all the functional genes and do away with the non-coding sequences, how the role of non-coding RNA non-coding?
Not do away. We cannot do away with the non-coding sequences because non-coding sequences are important and they are playing an important role in regulation. But you can identify sequences which are really not required because by deleting them, if the plant looks the same and does not lose any function, then you can say that this sequence was redundant. And this has been done actually. In case of microbes and also in case of yeast, they are trying to do it. You will have to find out. You see, by editing, you will have to delete a particular sequence and then you have to find out whether the function is affected. If the organism is not affected, then only you can delete it. Right. But non-coding sequences are very important. You can't survive without it.
So there is one Ajinkya who has asked why insect resistance breeding is less successful, especially in vegetables.
Well, maybe not enough effort. Yeah. That's what I can say. Not many people are doing it. You see, that's the reason.
Yes, sir. Sir, one question is on epigenetics. They wanted to have a brief brief summary on epigenetics, which is one of your favorite topics. But
I gave a lecture IRI last year on epigenetics. But epigenetics is a vast subject. We have recently published a paper in Plant Molecular Biology on histone methylation and its role in regulation of leaf rust resistance. Actually, I sent that paper to KV Prabhu, and he wrote me back a long mail for future experiments, which I can conduct. So he was excited about this paper.
Yeah, Gopal, you can inform that, those cast genomics lecture, they
Yes,
are on
sir.
YouTube, so they can watch that
Yes,
lecture.
sir.
So this presentation is on YouTube, and it will be available on this site, so it can be watched later also.
Okay, sir.
Anybody can write me an email if they have a question.
Sir, J.K. Moganty has asked what is the disadvantage of GWAS? If you can explain a little bit, it would be good. Yes.
I don't like GWAS. You see, the major problem with GWAS is that it is large number of false positives. Large number of false positives because it is based on LD. And LD is not based on linkage. So in many of the markers which we determine in GWAS, many of those markers therefore do not work. You see, I always quote an example that we invited proposals, research proposals on GWAS. 100 proposals were received and none of them was funded. Then Ashok asked me that I should write a review. And I wrote 2 reviews on association mapping published in Advances in Genetics. They are long reviews. So those who are interested in GWAS, they should read those 2 reviews. One is for the GWAS and the other is for post-GWAS, but after GWAS.
Sir, Geetika Bandari asks, can base editing mark the era of 2020s and beyond?
Oh, you never know what is coming next. But base editing, I think base editing is more precise in comparison to CRISPR-Cas. And base editing during the last 4 years has been improved. Even in 2020, there are a number of papers published on base editing. They are improving it every month. There is a paper for further improvement. I don't find so many papers on CRISPR-Cas, but on base editing, I think every month there is a paper showing further improvement.
Yes, sir. If any of the panelists would like to put any questions or discussion points to Professor P.K. Gupta, it would be they are also welcome.
Gopal, we
Yes,
like
sir.
to invite
Yes, sir.
Professor Bansal and then Dr. Dilan for
Yes,
their
sir.
brief, I would not say remarks, but they would like to say something on this presentation. Dr. Bansal?
Thank you, Professor Ashok Singh. It's an honor, actually speaking, I must say, to listen to Professor Gupta. And as I said in the beginning, informally, when we were talking, that he's a teacher to all of us. And we are proud to have him in Meerut in India, running a lab even now and publishing continuously papers. And not only research papers, also books and review articles. And today's lecture, of course, was again, kind of a lesson to me. I remember my courses attended in genetics division, IRI, doing PhD, and some of these, familiar slides and kind of work we stated was again refreshing my own knowledge, I would say this. And of course, there were several questions and he has covered all the aspects, but to me, what appears is that the kind of basic knowledge which is imparted will always remain, important for all the people, including those youngsters who are listening it, to take a note of it, that the basic understanding of the subject is the most important key. And in fact, that was one of the ideas also when we were developing this World Bank project, this National Agricultural Higher Education project, that there are excellent teachers, but then again, the students must imbibe those, good qualities of understanding and learning from the teachers. So this is, I'm sure, a very good initiative. Professor Ashok Singh, you have taken as Director IRI, Dr. Vishnathan, and many stalwarts, including VC. Last time also Professor Baldev Hillo was there as one of the panelists, or I was there as one of the participants again. And so what I see is it's a kind of a good dissemination of knowledge and re-emphasis to be again on the basic understanding of the subject, particularly for the youngsters, as you know, Dr. Gupta mentioned, you know, there were so many project proposals on association mapping, but none was funded because probably many of us did not understand that what must be the association mapping per se. You know, so there are various technologies available, but the fundamental basis of those technologies, the principles on which these technologies have been based, even for example, CRISPR, genome editing, base editing, you know, how are they being conceived and how can we then utilize them in crop improvement, I think, you know, therefore becomes important later. So I think Professor Gupta has covered, you know, all those basic aspects, discoveries, and it's a great lecture, kind of encyclopedia, right from genetics to genomics. I like the title very much, Mendelian genetics to modern genomics, and even beyond, people are asking those questions. So I'm sure there is a kind of great interest, and NAHEP, I'm personally pleased, is giving dividends, and I'm happy people like all Dr. Vishnathan, Dr. Manjaya, Dr. Madam Rashmi Agarwal, the Dean, Dr. Barnwal, I see many of them on the panel. They are doing some excellent job. Dr. Gopal Krishnan, you know, is there in genetics and all. I think they're doing excellent job. I'm very pleased, you know, to be here. And thanks to you, Professor Ashok Singh, for inviting me to give this opportunity today to as a panelist. Thank you very much.
Thank you, Dr. Bansal. May I request Professor Dhillon for his brief remarks. Dr. Dhillon, please. Sir, please unmute. Yeah.
Am I audible, please?
Yes, sir. Now it is fine.
Yes.
Yeah.
So thank you, Dr. A.K. Singh. Thank you very much for giving me this opportunity. And it's always a pleasure, a learning experience to discuss something or to listen. Dr. P.K. Gupta, storehouse of knowledge. He is one of the persons who is lucky to stay in science. He is one of the lucky persons to stay in science and then contribute to science and contribute to society through science. And that's how it was pleasure. I remembered my days, mid-60s, at Khalsa College, Amritsar, we were taught 4 Mendel laws. And when I came to PhD for MSc, there are only 2. So I spent one full day in the library to search. It is 1935 book by Sinnott and Dunn, where they mention 4 laws. So this is how science has developed, and Dr. Mansur, this is how we learn also. Of course, nowadays it is much better interaction. It's very
yes, sir, yeah.
And those are the days, even Mendel laws were not understood. And so how an old person like me, a new young person, will understand association mapping? Just see the confidence of Dr. Gupta. He has the confidence to say that I didn't know anything about association mapping. And that's what's unique about him. When you sit with him, storehouse of knowledge, starting from, I would say, Mendel, Galton, Fisher, and then coming to Buckler also he mentioned. That's a Buckler, just see the experiment which he is proposed and doing on B73. One of my paper was rejected in biometrics on this point that nobody will perform such a big experiment. And that is published in a Biometrical Journal, which is rated much lower than biometrics or biometrica. It is an estimation of gene difference in random mutant population. That was the only objection. But now Buckler, how the things change. And I will not say much. I will also say something about Dr. Chopra. You talked about him. Dr. Gupta, I have seen DNA only once as PhD student at IRI when Dr. Chopra just in the practical examination, he just showed us from the beaker on a, what we call, let us say, stick of shaker or something like that. Yeah. So only one, I have not seen the gene, what to say of gene, but I saw only DNA once, the thread. And that shows how the genetics has progressed. And I'm not in active science, so I leave it to the active science. Dr. Bansal is there, Dr. Shok is there, Dr. Viswanathan is there. So they will talk more, but I say that future belongs to molecular breeding. Molecular biology plus plant breeding. Very clear. It cannot be only sitting in the lab and doing synthetic this genome or gene editing, etc., etc. Phenotyping is going to be equally important in the field also. And that's why I'm very, very happy that you're writing a book on quantitative genetics. I'm very happy because that is what I'm missing. But I didn't understand. You said that quantitative genetics started in 1990. It was fashion 1960s.
Oh,
See,
no, no. I meant molecular.
yeah. You can say that quantitative genetics have been revived in 1990. I'm really very happy that you had done something which I wanted to do. So that's wonderful thing that you are doing. And then, Ashok, I have one request. In fact, I think I talked to my colleagues over here or to DDG Education, Dr. Agarwal. You have this project and we have a natural resource management. PhD is as much interested in your lectures and you must be as much interested in our lectures. If we can have some sort of permanent arrangement that all the lectures which you arrange, this should circulated, I am here and I am sure many colleagues of mine must be there because I circulated and I request them that please listen to this lecture because Dr. P.K. Gupta at the moment may be the most outstanding working scientist, most outstanding working geneticist or molecular geneticist or molecular cytogeneticist, all the things put together. And we have so many things to learn from him. So if we can work out something permanently, we can share what
we will do that.
Natural resource management and what you are doing in this. I don't know what is the title, but I know the area in which, yeah, Dr. Bansal was very helpful to both the institutes when he was in ICAR headquarters. So thank you, Dr. Sir. Thank you very much. I will not say much in science. You see, now it is my 10th year. I wrote science 9 years ago, and I say that my genotype has changed after I joined as Vice Chancellor. So I will not touch science.
Oh, it happens.
Thank you very much. It was a pleasure. And thanks to Ashok for this support.
And it was pleasure to have you on this panel. Any other panelists who would like to say something, or if you have any query to Professor Gupta, please feel free. We have still time. I said Professor Gupta that we will be there till 5 o'clock, so we have 20 minutes to go. If you have any question from panelists, anyone?
If you can take questions from this.
That?
Can take question from the participants because many students they are asking questions if they can be allowed to take.
No, see, the participants problem is that they are saying that they do not know how to unmute and they are not able to speak. That's why they are giving the comments. So if you can let them know how they can unmute themselves, then they can ask the question.
Coming, sir. Some students are coming. One Shailesh,
can you go ahead, unmute yourself and ask the question? Unmute yourself.
Dr. Roy, can you give them a clue how to unmute?
Sir, I have already allowed them to talk, so now it is up to them to unmute at their own system.
Okay.
They have been allowed to talk, sir.
Okay. So seems there is some technical hitch. All right. I mean, there is a lot of
yes, sir. Arjun can talk now.
Yeah, Arjun.
Good evening.
Good evening.
Sir, I wanted to know, like, what is the prospect of GPS? Actually, presently I'm working on GPS, but not sure, like, how much it is prospective in the future. I'm working on Chile for GPS. Can P.K. Gupta sir shed some light on GPS?
So GPS, you are talking by sequencing?
Yes.
By sequencing, there are a number of vendors who are doing it.
Actually, I'm working on DD-RAD, sir, at present.
You are working on?
On DD-RAD, GPS on DD-RAD technology. Okay. Double digestion.
Okay.
You just shed some light on that, sir. Like,
so ultimately, what is your objective?
Actually, I'm working on finding the gene for one trait, for a disease trait in Chile, sir.
So genotyping, if you want to do genotyping, I think particularly for association mapping or even for interval mapping, I think GBS is one of the cheapest and there are a number of vendors who are prepared to do it. Normally, I think for 100 or 200 accessions, they would take about 6-7 lakhs. That's all. You have not contacted any vendor in this connection? Hello? Can you hear me?
He's muted again, muted himself. There is some from Shakti. Shakti, can you go ahead with your question? Unmute yourself and go ahead with your question. Shakti Muller.
I think anybody has a specific question can write an email to me. Gopal, you have any questions in the chat box?
Yes, sir. There are 115 questions. I am trying to go through it, and yeah, sir. There is one question from Vishwas. He asks, "What is the future of conventional breeding in the era of molecular breeding?"
Oh well, conventional breeding continues. Molecular markers only supplement. I always talk about it. One of the reviews on molecular markers which I wrote, the title was Marker Assisted Selection as a Supplement to Plant Breeding, Conventional Plant Breeding. And at many places I suggested, at Karnal also I suggested, biotechnology should be merged with plant breeding.
Absolutely, sir. I think they should be going through the.
It's all plant breeding at the end of the day.
Just hold on. There is a question on phone.
Okay, sir.
Yes.
Yeah.
Yes.
Now, all of us are targeting for improving the biotic cells or abiotic cells or quality.
Right.
Say, for example, we have the genetic potential. Already we have a X, Y, Z genotypes are there.
Okay.
They already have the genetic potential. Say, for example, in rice, it is something like 3.5 ton. Can you consider 3.5 ton is the best potential genotype right now?
Rice you are talking about?
No, rice. Say example. I am taking example. Just say.
Ashok should answer that question.
No, this is not a question to Dr. Ashok. Sir,
No, you see the biotic and abiotic stresses both can be handled through molecular markers. There is no question about it. You have to find out. Yes, say it again.
Our question is, it's not only biotic abiotic stress, because to meet the demand by 2050,
okay.
2050, is it only biotic abiotic stress enough, or we need another quantum jump in yield? Can you target most of the yield QTLs with different methods? But what we are right now targeting is we are always talking on many things on biotic stress management.
No, but
we are not talking on yield jump. This is my question. So you can please highlight what to do for yield jump, another jump, like in what has happened in
but when you are talking about abiotic stress, then also you are talking about yield under stress.
Yield under stress, yes, sir.
We are always talking about yield. You see, whether biotic or abiotic, you can't talk about yield without talking about the different stresses which the crop faces. So you have to, whenever we study, we want to find out QTL for drought, we take yield under drought condition as one of the important parameters. So yield is always there. And yield per se does not have any gene. Yield consists of so many components.
Okay, that's fine, sir. But what I want to know is, do you need one more quantum jump in most of the crop plants?
If I can come in, sir.
Yes, please.
Yeah. So your question
now,
is, can you? I think he should be able to listen me. Your question is how we can get to new yield jump, like from the time of green revolution in rice and wheat. So what Professor Gupta described is about, the genomic selection. Now, genomic selection is a tool to accumulate the favorable alleles or genes which are scattered into population together. And this accumulation will certainly bring in a quantum jump. And that's what the approach is. Now, one you have is genetic gain, another is protection of genetic gain or realization of genetic gain. To realize genetic gain that you have achieved, you need to have tolerance to biotic stresses, tolerance to abiotic stresses. Unless you have this, you cannot realize the gain, genetic gain that you have achieved. But to increase the genetic gain further for yield per se, you need to find many more useful genes which could be present in land races, wild and weedy relatives, and bring them into the cultivated pool. And that is what the approach is. For example, from cultivated species and from land races, the genomic selection is one approach to bring in favorable genes, and the wide hybridization, you had advanced backcross QTL methods. These are the approaches to bring in new genes. So that will definitely help in improving the genetic gain further to have a quantum jump. Like new targets, for example, in case of rice, our best yield realized is 10 tons per hectare, best yield. But the average yield is about 3.5 tons per hectare. Now this gap of 3.5 tons to 10 tons per hectare is something to manage with, whether it is biotic stress, abiotic stress, all these things have to manage to maximize this. And to go beyond that, you have to plow in new alleles and new genes. I hope I have addressed your question. Is not connected, I think.
Yes, sir.
Yeah.
Yes, sir. Sir, there is one question on the agriculture education system. Being a thorough teacher throughout his life, there is a question from one of the participants, Vishnu Kant. He asks, how can we improve the agriculture education system? And do you think we still need to empower practical oriented teaching? And how can we achieve this?
Oh, well, it's not a easy question. I delivered a lecture on teaching of genetics. You should listen to that lecture. It is available on YouTube even today.
Okay,
teaching of genetics.
It was delivered in IRI only in BP Paul Auditorium. Yeah.
Yes.
Another question by one of the participants who is asking, like, we edit a gene and we keep on editing genes and genes and genes. After some time, the genome itself becomes completely different from what it was earlier. It leads to complete genomic change. What will happen that way, like in the long run? That is the question.
I think he is overestimating the changes we bring about. The changes which we bring about are just negligible in comparison to the whole genome. We are not tampering with the whole genome. We are not tampering with the whole genome.
What will happen that way, like in the long run? That is the question.
I think he is overestimating the changes we bring about. The changes which we bring about are just.
Sir, you have to switch off your mobile. I think it is echoing. Yeah. Yes, sir.
Rashmi wanted to say something.
Rashmi has to propose vote of thanks, so she is waiting for her turn, I guess.
I will say, sir, when I will propose a vote of thanks.
Sir, one last question. One last question by Biswajit Lenka. But why is it said they are mostly biallelic?
Biallele?
Yes, sir.
Say it again.
Sir, SNPs can have 4 different bases as alleles, but why?
Yes. No, but there are examples where you have more than 2 alleles.
Yes, sir. In case of SNPs, yes, sir.
There are examples where there are more than 2 alleles.
And Anil is asking what should be the optimum population size?
Population size for what? For association mapping or for what?
Hello. Optimum population size for association mapping, sir.
More the merrier.
Yeah.
I think we can't afford. Otherwise, I think we should have about 1,000 taxations.
Yes, sir. There is one more interesting question, sir, from Asif. He is asking, is genomics contributing for future green revolution in India?
Oh, well, B.S. Yadav has written a paper in Current Science that we are having green revolution one after the other. We did not stop it. So we are in the era of green revolution, wheat production, rice production increasing almost every year. So we are in the process of green revolution. We did not stop it.
Yes, sir. Thank you. Sir, Prashant Chingote asks, if one wants to characterize the trait that has not been characterized at molecular level yet, but identified phenotypically, what will be the best method to characterize it in an orphan crop with least funding resources?
Oh, that is a big question. He wants me to prepare a project for him.
Yeah, you said it. You have to prepare a project proposal and then plug in the gaps so that you can get into. Yes, sir. Sir, you please. AK Singh, sir.
Yeah. Gopal,
Yes,
I am
yes,
there, and
yes,
I think we are now close to the time that we have scheduled for, and this has been a great experience. The journey from genetics to genomics, covering all aspects. It was a treat, like closing eyes and just listening, like such a pleasure to listen to Professor Gupta. I think what he has described, the dates and events should be memorized by each and every genetic students, like you do Hanuman Chalisa or something of that sort. That should be the spirit for the students to understand and to establish connectivity. Sir, you are talking about association mapping and the efforts that you started by writing those reviews, and the first efforts that you initiated in DVT. 2-3 rounds of meetings were held, and then the projects were sent to Ed Buckler, and then finally it did not materialize. But now you will be pleased to know that the DVT has funded at least 4 major projects on genome-wide association mapping, and particular, yes, they are in the millets, oilseeds, and rice and wheat. The one project that we are having on rice is about 50 crores plus, and wheat one is also about 40 crores. And there we have now started with 15,000 land races, unique land races, name land races, all 15,000, and we are going to do resequencing and extensive phenotyping. They have been all planted. So what you thought about, I think 7-6 years back, is coming true now, and I think soon we will have better output.
We grow with time. We grow with time, you see.
Right, sir.
Many of the things I talked about, I didn't know when I was a student. I learned it all afterwards.
So it has been a great pleasure, sir, and I request Dr. Rasmi for a vote of thanks. Dr. Rasmi.
Thank you, sir. Can you all listen to me? It's my really pleasure and privilege to propose a formal vote of thanks to Professor P.K. Gupta. Sir, I know I have met you earlier also, and you are really, really outstanding teacher, a researcher, an academician who has really given us such a treat to our brain that all through Mendelian genetics to modern genomics, you have highlighted each and every step. And I must say, sir, not only you have just given a historical account, but you have correlated and given a scientific basis of it, how we have progressed. And also, sir, the understanding which you gave us for the Mendelian genetics, that was really so appreciable, that what were the lacunae in his experiments and how we can relate now to the modern genetics and genomics, sir. And this last part of your talk on base editing and genome editing, that really is an input for the young generation to go for application part of it. Of course, the breeding and the conventional breeding, one cannot stop, But using these modern tools, we can cut short the period and do it, sir. All these you have highlighted so well, sir. I'm so proud and privileged, sir, having interacted with you on.
Dr. Rasmi, you're muted. Kindly unmute. Dr. Rasmi, please unmute yourself.
Sir, I'm so thankful, sir, to you that I could publish a review article with you on the biology of bipolaris, sir. Really, it was a wonderful experience. And after that, we are doing a lot of work in same direction, sir. So all of us are so much thankful to you on behalf of Director IRI, on my own behalf, and on behalf of NHEP CAST team. We are so thankful to you, sir, and we have been listening to you earlier also, and so many occasions. And this had been a really great lecture, sir, outstanding lecture. We all, like, it's a standing ovation to you on this lecture, to all, not only to the geneticists, breeders, students, faculty, but also to all of us, like me and many other like me in the faculty. So, so much thankful to you, sir, for sparing at this age, at this stage. And you are really a role model and inspiration to all of us. I'm so much thankful to Dr. A.K. Singh, our director, honorable director, and he is organizing such nice webinars. And we are really enriching our knowledge, and all the students and faculty are also getting benefit out of it. Sir, thank you so much, sir, honorable director IRI, for giving this opportunity to all of us. And a special thanks to Professor K.C. Bansal and Dr. B.S. Dhillon for coming. These are our special invited, distinguished invitees who joined this webinar and gave their input and their time also. Thank you so much, both of you. And thank you to Dr. Vishwanathan, NHEP CAST team, all the core members, Dr. Gopala, and many faculty of IRI and others, and more so the students. Sir, NHEP CAST is student-oriented program, and we see more than 1,000 participants are there. So it's a wonderful experience for the students also. And I'm sure they are all enlightened, and they will have large areas to work on and to read and do a lot of things. So thankful to each one of you who really helped in organizing, Dr. Roy Burman and others who organized this webinar. And series of webinars are being organized by Dr. Vishwanathan and NHEP program. This NHEP program is a very nice program, World Bank funded program by our ICR Education Department. So we are all thankful to our DDG Education in ICR for funding this program, and to the World Bank for giving this opportunity to all of us to listen to such, sir, eminent speakers like you, sir, researcher and speaker. Sir, thank you so much. I will again get some opportunity to talk to you about my work on that bipolaris, sir. Definitely we discuss, sir. Thank you, each one of you, and good evening, sir. Good day.
Thank you, sir. Thank you so much. Thank you very much. We will, yes, sir.
Thank you, sir. We can now leave, sir.
Thank you, everybody.